Fragments of a biblical scroll, along with other relics, have been found in desert caves in Israel.
For 2,000 years, these scrolls were believed to be the voice of one ancient group, a single, isolated desert community writing scripture in silence. But DNA just proved that idea may be completely wrong. What if everything we thought about the Dead Sea Scrolls, every theory, every textbook, every museum exhibit, is built on a mistake? Not a small mistake. A fundamental one. A mistake that changes how we understand the Bible itself. This is not speculation. This is not a fringe theory. This is what the science actually says, and scholars were not prepared for what came next. In early 1947, a young Bedouin shepherd named Muhammad Eddib was searching for a lost goat in the cliffs above the Dead Sea. He was not an archaeologist. He had no training, no equipment, no mission. He was just a boy looking for an animal in the desert. He threw a rock into a dark cave opening. He heard the sound of clay pottery breaking, and he climbed inside to see what he had hit. What he found would become the greatest accidental discovery in the history of archaeology. Inside that cave, wrapped in linen and sealed inside ancient clay jars, were scrolls, manuscripts, documents so old that the world they described had been buried under centuries of dust and silence. That single moment, a boy, a rock, a broken jar, opened the most important window into ancient religious history the modern world has ever seen. Over the next decade, between 1947 and 1956, researchers and Bedouin explorers uncovered manuscripts in 11 different caves near the ruins of Qumran, on the northwestern shore of the Dead Sea. What they found staggered the scholarly world. The Dead Sea Scrolls contained over 200 copies of books from the Hebrew Bible, the oldest surviving evidence for the biblical text anywhere on earth. Before this discovery, the oldest known copies of the Hebrew Bible dated to around 1000 CE. The scrolls pushed that date back by a full 1,000 years in a single find. A millennium of uncertainty collapsed in a moment. But hidden inside the parchment itself, at a level no human eye could see, something was still waiting. Something nobody thought to ask about for another 70 years. The scrolls did not come to scholars whole and intact. Decades of decay, animal damage, and fragile parchment had reduced the collection to over 25,000 individual fragments. 25,000 pieces. No clear guide for how they fit together. No complete picture on the box. Scholars spent generations sorting them by handwriting style, by ink composition, by the visible patterns of the text itself. Progress was slow. Arguments between experts about which fragment belonged to which scroll were common, sometimes bitter, and never fully resolved. A single fragment might be assigned to one scroll by one generation of scholars and reassigned by the next. The puzzle kept shifting. The picture kept blurring. And beneath all of that, beneath the ink, beneath the handwriting, beneath every argument anyone had ever had about where these pieces belonged, the answer had been sitting there the entire time. Written not in ancient Hebrew, not in Aramaic or Greek, written in DNA. In 2012, a molecular biologist named Oded Richavi and a biblical scholar named Noam Mizrahi sat beside each other on a bus during an orientation program at Tel Aviv University. They were strangers. They worked in completely different fields. One spent his days reading genetic code. The other spent his days reading ancient scripture. But as they talked, they realized they shared the same problem from opposite directions. Richavi had tools for reading biological information encoded in DNA. Mizrahi had thousands of fragmentary scroll pieces that no one had been able to fully organize. And then one of them said something that neither had considered before. The scrolls were made from animal skin, processed into parchment. And animal skin contains DNA. DNA that does not simply vanish because an animal died 2,000 years ago. Ancient DNA degrades, yes. It fragments into short, damaged pieces. But under the right conditions, the extreme dryness of the Judean desert, the stable temperatures of sealed limestone caves, traces of it survive. The insight was deceptively simple. If you could extract and read that ancient DNA, you could identify which fragments came from the same animal. And if they came from the same animal, they almost certainly came from the same scroll. You could solve the puzzle using genetics instead of guesswork. 8 years after that bus ride, they had built the technique to do it. The method they developed was precise enough to extract real data without destroying the manuscripts themselves. Researchers scraped microscopic amounts of scroll dust from the uninscribed backsides of the fragments, the blank side, away from the text. The precious writing itself was never touched, never put at risk. Using a technique called PC, polymerase chain reaction, they amplified those trace amounts of ancient DNA to detectable levels and ran them through sequencing machines. The samples contained two kinds of genetic material mixed together: modern human DNA left by everyone who had ever handled the scrolls without gloves, and fragmented ancient animal DNA from the original parchment. Separating those two signals, filtering contamination from data, modern from ancient, was one of the most technically demanding challenges the team ever faced. The ancient DNA extraction itself was carried out under extraordinarily strict laboratory conditions at the paleogenomics lab of Professor Mattias Jakobsson at Uppsala University in Sweden. Researchers wore full-body suits. Every surface was controlled. Every breath was a potential contaminant. And they pulled it off. Once the ancient DNA was sequenced and cleaned, sophisticated computational tools reconstructed genetic relationships between fragments. They could determine whether two pieces of parchment came from the same individual animal, from closely related animals of the same flock, or from completely separate populations in different geographic regions. Professor Jakobsson described the achievement as genuinely remarkable, given that the parchment had been processed, used in a rough desert environment, sealed in a cave for 2,000 years, and then handled without gloves by modern scholars for decades after that. And once they had the data, what it revealed startled the entire field. The very first result was already surprising. Every fragment tested was made from sheepskin, except for two. Just 2 fragments, out of the entire collection tested, were made from cowhide. Before this research, no one had definitively established which animal species had been used to produce most of the scrolls. Scholars assumed sheep or goat based on historical context, but they had no genetic proof. Now they did. The dominance of sheepskin made sense geographically. Sheep were the primary livestock of ancient Judea. They were the most available animal to a desert community. But those 2 cowhide fragments, those 2 exceptions, were about to rewrite history. Both cowhide fragments belonged to the book of Jeremiah. This single detail changed 70 years of certainty. Here's why. Cows cannot survive in the parched Judean desert. They require large amounts of water and grass, resources the barren landscape around Qumran simply cannot provide. So whoever produced the parchment for those Jeremiah fragments did not live at Qumran. They could not have. They lived somewhere else, somewhere more northerly, more fertile. Somewhere like the area around Jerusalem itself. The DNA had just proven that at least part of the Qumran library was not produced locally. It was brought in from the outside world. And then the implications kept growing. One of those cowhide fragments had previously been assumed, based entirely on visual and textual analysis, to belong to the same scroll as a sheepskin fragment. Scholars had accepted this conclusion for years. It was in the literature. It was treated as established fact. The DNA dissolved that assumption completely. The 2 fragments were made from different animals of different species. They almost certainly came from entirely separate scrolls, produced in separate locations. A scholarly conclusion that had stood for years was overturned by a single genetic test. Then came something even more profound. The DNA study confirmed that 4 distinct copies of the Book of Jeremiah were represented among the Qumran fragments, and each reflected a different version of the text. Not different handwriting, not different ink, different content. Today, a Torah scroll in Jerusalem is virtually letter for letter identical to one in New York, in Sydney, in Lagos. The very idea that variation in the sacred text would be acceptable is almost unthinkable in modern Jewish or Christian practice. Scripture is scripture. The words are fixed. But at Qumran, different versions of the same biblical book, with genuinely different content, were sitting side by side, treated as equally authoritative scripture by the same community at the same time. This is not a minor textual variant. This is a fundamentally different understanding of what scripture was. In the era when both classical Judaism and early Christianity were taking shape, the argument was not about which precise wording of Scripture was sacred. The text was still being negotiated, still being shaped. The process of settling on a single standardized biblical text was a later historical development, happening around 100 CE, not something that was fixed from the moment of composition. The DNA study confirmed this not just as a textual theory but as a physical, biological fact embedded in the very skin of the manuscripts. The Bible as we know it was not handed down complete and unchanging. It was assembled, negotiated, and standardized over centuries, and the Dead Sea Scrolls are the proof. Among the more quietly astonishing findings was one involving a liturgical text most people have never heard of, the Songs of the Sabbath Sacrifice. The Dead Sea Scrolls contain 10 copies of this single non-biblical composition. 10 copies. That is more than the number of surviving copies of some canonical biblical books found at Qumran. Clearly, this was a text the community returned to repeatedly and considered deeply important. Scholars knew it mattered. What they did not know was how far it had traveled. Fragments from 3 of those copies, found in 2 different Qumran caves, were made from the skins of closely related sheep, suggesting they were produced by the same local community. But a 4th copy had been found somewhere else entirely. Masada, the famous desert fortress approximately 55 kilometers south of Qumran, where Jewish rebels made their last stand against Rome in 73 CE. Scholars had long debated whether that Masada copy had been brought there by refugees fleeing from Qumran after the community's destruction. The DNA settled the debate. The Masada sheep came from a genetically separate population. The scroll was independently produced elsewhere. The Songs of the Sabbath Sacrifice were not a quirky specialty of one desert sect. They were known, valued, and copied by Jewish communities across a much wider geographic area. No one had suspected the extent of that world until the genetics showed it. And this discovery carried a second consequence. For most of the 20th century, the dominant theory was clear and confident. The Dead Sea Scrolls were written by the Essenes, an ancient Jewish sect that established a monastic community at Qumran in the mid-2nd century BC. They lived apart from mainstream Jewish society. They devoted themselves to scripture, manual labor, and prayer. They harbored apocalyptic visions of a final confrontation between the forces of light and darkness. The scrolls themselves seemed to support this portrait. It was a clean story, a compelling one. The DNA research shattered it. By showing that multiple scrolls came from outside Qumran, produced from animals that could not have lived in the desert, the study proved that many documents were deliberately collected and brought in from other communities across Judea. The library was not the private writings of one isolated sect. It was something far broader. A snapshot of the full diversity of Jewish religious thought across an entire region, in the century before and during the time of Jesus. Which makes it even more historically significant than the Essene theory alone ever implied. Among the quietest findings of the study, and perhaps the most haunting, is one that deserves far more attention than it has received. The genetic signature of one particular Isaiah fragment does not match any of the known Qumran cave populations. It came from somewhere else. Somewhere that has not been identified. Somewhere that, as of this moment, no archaeologist has ever found. The DNA evidence points to the potential existence of an additional site, another cave, another collection, another deposit of ancient manuscripts, that is still out there, waiting in the dark. The Isaiah scroll already holds a celebrated place in the Dead Sea Scrolls collection. The Great Isaiah Scroll from Cave 1 is one of the best preserved ancient biblical manuscripts ever discovered, stretching over 7 meters and containing the complete book of Isaiah in pristine condition. But this anomalous fragment is pointing somewhere no one has looked. The region around the Dead Sea is vast. The caves are numerous. The same conditions of extreme dryness and stable temperature that preserved the Qumran scrolls exist across a wide area of the Judean desert. Somewhere in those cliffs, it remains genuinely possible, more than possible, that clay jars are still sitting in the dark, holding documents that no human being has read in 2,000 years. The evidence for undiscovered scroll sites does not rest on that single fragment alone. In February 2017, A 12th cave was discovered on cliffs west of Qumran. No scrolls remained inside, but archaeologists found pickaxes and empty, broken scroll jars. The cave had contained scrolls. They were looted almost certainly in the 1950s. Those scrolls may now be sitting unidentified in a private collection somewhere, never connected to their origin, never recognized for what they are. In 2021, Israeli archaeologists working in a site dramatically named the Cave of Horrors recovered additional fragments containing text from the books of Zechariah and Nahum, written in Greek, proving that new scroll material was still surfacing 7 full decades after the initial discoveries. The desert had not finished speaking. It had only paused. And the DNA method adds yet another dimension to the search. Since the forgery market for Dead Sea Scroll fragments has long attracted criminals, in 2020, the Museum of the Bible in Washington, D.C. was forced to announce that all 16 of its Dead Sea Scroll fragments were modern forgeries, acquired through the antiquities market and displayed as authentic for years. Authentication has become as urgent as discovery. Genuine ancient fragments carry ancient animal DNA that matches the genetic profiles of sheep populations from ancient Judea. Modern forgeries carry DNA from entirely different animals with entirely different genetic characteristics. A single test can now separate the real from the fabricated in a way that decades of visual inspection never could. The coming decade of Dead Sea Scroll research may be the most exciting since the 1950s. The Dead Sea Scrolls were not the product of one sect. They were not the voice of one isolated community writing in the desert, waiting for the end of the world. They were the collected evidence of an entire religious world, diverse, contested, alive, still in the process of becoming what would eventually be called the Bible. The DNA did not just help organize fragments. It dismantled a century of assumptions. It showed us that the text we call sacred was shaped by human hands, copied by multiple communities, negotiated across generations, and standardized only after centuries of variation. It showed us that the boundary between one version and another, between one tradition and the next, was never as fixed as we imagined. It showed us that history itself was never as settled as we believed. And it showed us something even more extraordinary than any of that. Somewhere in the Judean desert, in a cave no one has entered, in jars no one has touched, in darkness that has held its silence for 2,000 years, the story is not finished. The genetic compass is pointing. The surveys are moving. The technology is advancing. And somewhere, perhaps just beyond the edge of what we currently know, manuscripts are waiting that will force us to rewrite the story again. The Dead Sea Scrolls did not just preserve history. They revealed that history itself was never as fixed as we believed. They are still speaking. We are only just beginning to hear them.